Inside the human ribcage lies a dense, spongy, and highly compartmentalized matrix. These paired organs serve a single physical purpose, acting as the site where oxygen from the atmosphere enters the systemic bloodstream and carbon dioxide is extracted. Human pulmonary anatomy is defined by strict spatial compromises, hierarchical segmentation, and specialized circulatory systems. To understand this system, we will trace the anatomy from the macro constraints of the thoracic wall down to the thin epithelial layers where gas diffusion occurs. This structural hierarchy allows the respiratory system to maintain function even during localized physical or pathological damage. The lung functions as a highly optimized biological engineering solution, partitioned into segments to ensure that localized failure does not trigger a total collapse of gas exchange. This diagram compares the physical profiles of the two lungs. The right lung is significantly heavier, shorter, and wider than the left. It is divided into superior, middle, and inferior lobes, separated by the oblique and horizontal fissures. The left lung possesses only two lobes, a superior and an inferior, divided by a single oblique fissure. It also features a sharp indentation called the cardiac notch and a small projection known as the lingula. This asymmetry is a direct result of the mediastinum's contents. The human heart is displaced toward the left side of the chest cavity, dictating the space available for lung tissue. The left lung sacrifices a third lobe to accommodate the cardiovascular system. This gross anatomical asymmetry is a mandatory spatial compromise required to house the heart. On the medial surface of the lung lies the hilum, a vertical slit where all vessels and airways penetrate the organ. The structures passing through this slit form the root of the lung, comprising the main bronchus, the pulmonary artery, pulmonary veins, bronchial vessels, and lymphatics. These structures follow a strict spatial arrangement that can be mapped using the mnemonic RALS. RA denotes the right lung, where the pulmonary artery is positioned anterior to the right main bronchus. This layout places the eparterial bronchus, a specific branch of the airway, directly above the pulmonary artery on the right side. LS denotes the left lung, where the pulmonary artery is situated superior to the left main bronchus. This topography prevents the rigid airways from compressing the low-pressure pulmonary veins and provides predictable landmarks for thoracic surgeons. Removing the soft lung tissue reveals the rigid cartilaginous framework of the bronchial tree. This diagram illustrates the bifurcation where the trachea splits, showing a distinct geometric difference. The right main bronchus is wider, shorter, and vertical. The left is narrower, longer, and angled more horizontally to bypass the heart. Because of this vertical alignment, aspirated foreign bodies, such as food particles, travel down the right main bronchus significantly more often than the left. From these main stems, the tree undergoes fractal division. Main bronchi split into lobar bronchi, then segmental bronchi, and finally into terminal and respiratory bronchioles. These architectural angles dictate the distribution of air and the likely path of inhaled pathological threats. Each lung is composed of bronchopulmonary segments, functionally independent units separated by connective tissue. There are 10 distinct segments in the right lung and 8 to 10 in the left, each supplied by a dedicated bronchus and artery. This modularity allows for the surgical resection of a diseased segment without compromising the air or blood supply of the surrounding healthy tissue. While air is distributed through these segments, the lung relies on a dual circulatory system to manage blood flow. Functional circulation is provided by the pulmonary arteries, which carry deoxygenated blood to the alveoli for gas exchange. Nutritional circulation is handled by the bronchial arteries, which provide oxygenated blood to the structural tissues and the visceral pleura. This redundancy in vascular routing and segmented isolation prevents localized damage from leading to systemic respiratory failure. Descending to the absolute terminus of the bronchial tree leads to the alveolar sacs, where the physics of gas exchange occurs. The walls of each alveolus are primarily composed of type 1 pneumocytes. These are flat, simple squamous cells creating the thin barrier required for gas diffusion into the capillaries. Scattered among them are type 2 pneumocytes, which serve a specialized chemical role. They secrete surfactant, a fluid-reducing surface tension, which prevents the microscopic alveolar walls from collapsing during expiration. Alveolar macrophages, or dust cells, patrol the space to consume microbes or debris that have reached this depth. The macroanatomical features of the lungs, the asymmetric lobes and the rigid bronchial tree, exist to support and protect these simple squamous cells. To move air, the diaphragm contracts and flattens. This expands the thoracic cavity, creating the pressure gradient necessary to draw air inward. Expiration is a largely passive process, driven by the natural elastic recoil of the lung tissue as the thoracic volume decreases. This continuous movement against the ribcage requires a frictionless interface, the pleura. This serous membrane consists of a visceral layer covering the lung and a parietal layer lining the thoracic wall. The potential space between these layers contains lubricating pleural fluid, which eliminates mechanical friction during the breathing cycle. Respiration integrates macromechanical shifts in thoracic pressure with molecular diffusion across the alveolar membrane.